Hello, Dr. Kong. This is Johnny Macaroon doing the Mutations in Drosophila melanogaster project presentation for the fall 2022 semester. Now, a little bit about the background of this presentation is that we will be studying the passing down of mutations over 2 generations from what is initially called a P-cross or the test cross, of which each fly is homozygous recessive or dominant. We'll discuss that in the hypothesis. The mutations that we were analyzed were vestigial and ebony, which are both recessive traits, which follow the law of independent assortment, meaning that these genes are separated independently from one another. This follows a dihybrid cross with a chi-square goodness of fit analysis to determine whether observed ratios meet expected ratios and how well they fit one another. The purpose of the project is to breed the fruit flies and analyze how Mendelian inheritance for sex-linked and autosomal genes are present over multiple generations, in this case 2. Our group will deal with vestigial and ebony mutations. In understanding mutations in Drosophila melanogaster, scientists can extrapolate an understanding of sex-linked mutations and mutations in independent assortment, otherwise known as autosomal mutations. This experiment will allow us to understand that the phenotypes in wild-type biota are dependent on the genes that are passed down generationally. For the methods, we will observe vestigial wings and ebony bodies, which are not sex-linked. They are a product of independent assortment. And our first batch of flies, known as the parental cross or the test cross or P-cross, was given to us on October 13th of 2022. Our F1s were observed on the 21st of October and our F2s on the 28th. Initially, before flies can be observed, part of the method is to create an environment in which the flies can survive and breed. This was done by obtaining a vial, an empty vial, pouring the food in, and pouring the water in so that they can survive and breed in a controlled environment. In order for the flies to breed after the environment has been set up, they must be anesthetized with fly nap, and upon their anesthetization, we can analyze the flies while they're asleep. Now, what's important to note when observing the flies is how you can tell the difference from one fly or another. The wild type have round red eyes and regular colored bodies, very common colored bodies. They're found in nature. The male flies have sex combs, while the female flies do not. And in some cases, while under the influence of fly nap, females can even be seen laying eggs, which makes it very obvious to determine their sex. Now, the results for this experiment, after analyzing the F1 and F2 generations, are present in the group F2 chart. Now, a chi-square analysis was done on the F2 chart to show the statistical goodness of fit of the distribution of mutations and genes throughout the experiment. The reason we do the F2 is because they're the final product of all the breeding and variation. For our group results, we were expecting a 9:3 to 3:1 ratio of approximately 300 wild type, 100 ebony body, 100 vestigial wings, and 34 double mutants for ebony and vestigial. But our observed data was a little different. That will be explained by the chi-square value in just a moment. The wild type observed were 336 with 81 ebony body, 92 vestigial wings, and 24 ebony and vestigial. After calculating the expected versus observed ratios and doing the chi-square analysis, you can see we have a final value of 11.51 with 3 degrees of freedom. That is a rather high chi-square number when compared to the chi-square chart. It rejects the null hypothesis. Therefore, our flies did not have, the results were not as expected. But if you look at the data, it's not too far off from what was expected to say that the experiment was a failure, because it was not. Thank you. The values here show that the spread was not too far off as expected, but far off enough to be statistically significant. Excuse me. Our hypothesis indicates the possibilities of the P genotype, the parental genotype. After analyzing our F2 flies and seeing a picture of the F2 flies right here. After analyzing our F2 flies right here, we did reverse inferences. We worked backwards to determine the possible genotypes for the parental flies. The only possible P cross is homozygous wild type vestigial with homozygous ebony wild type, and so basically the possible P cross is homozygous wild type for ebony and vestigial, and homozygous recessive for vestigial and ebony. Now, as mentioned earlier, we needed to see a 9:3:1 ratio, in which our case, the data suggests we did not. The crossing scheme. According to the crossing scheme and the 9:3:1 scenario, for every 16 flies, approximately 9 should be homozygous dominant, 6 will be heterozygous, and 1 will be homozygous recessive. And as mentioned, our group produced generations of flies that did not meet this ratio, therefore our null hypothesis was rejected. The result of the experiment, with 3 degrees of freedom in the chi-square analysis, was that the null hypothesis was rejected. In other words, group results were not as expected. And in reflection of why there was miscommunication, several flies died, several flies weren't accounted for. So human error, along with the randomness that comes with breeding, gave us a result that was not as expected. And that is mentioned in the discussion section here. Observed results do not support the hypothesis. Miscounting, imbalance, or P1 or F1 genotypes, and randomness. This pattern of inheritance was an unbalanced yield of wild type versus vestigial versus ebony versus double mutant. Although the results were not too far off from having an expected yield, wild type flies were overrepresented while the corresponding mutations were underrepresented. Now, troubleshooting and learning reflection.
Don't go too fast. Okay.
The deviation is possibly due to the mistranslations in communicated data among the group. Randomness is also a factor, but the flies were counted to the best of our ability. Although the values were high, they followed a somewhat consistent representation. They were mostly wild type with relatively equal distributions among single mutants, with double mutants being the least represented population of the group. If there were any suggested changes to the experiment that I would make, it's that there should be more focus on the Fruit Fly Lab manual. The manual I found was very helpful. It provided a lot of useful information, such as how to culture the flies, how to use the Flying app, how to count your flies and observe them and analyze the genes. I found the Fly Lab manual to be incredibly helpful. And I think that there should be more of a focus on it. And acknowledgements for my amazing group members and you, Dr. Khan. Your class was amazing. I had a great time. I couldn't stop talking to my friends about it. It was truly a blessing being in your class. Thank you.
Thank you very much, Doctor, for your patience and for waiting for Johnny to recover. I'm going to try to edit the video so that you can have a good view. God bless. Thank you.